Okay, welcome to lecture 12 where we will be talking on gluconeogenesis. Now, before you listen to this lecture, I will ask you to go through the lecture on glycolysis. The reason being, gluconeogenesis is reverse of glycolysis. So if you do not listen to the lecture on glycolysis, and understand the inherent concepts of glycolysis, it will be very difficult for you to follow the concepts delivered in the lecture related to gluconeogenesis. In this lecture, first we will define what is gluconeogenesis, and the definition is very important. You need to understand the basic idea behind the definition. Secondly, we will look at the irreversible steps in glycolysis which are made reversible in the process of gluconeogenesis. And this is the reason I stressed in the first slide on why you should listen to the lecture on glycolysis before you go through gluconeogenesis. We will talk a great deal about the regulation of gluconeogenesis by the regulation of the enzyme Pyruvatecarboxylase, and you have been introduced to this enzyme during the tutorial session where we talked about the conversion of pyruvate to oxaloacetate. We will then talk about futile and Cori cycle, which operates between the liver and the muscle, and we will focus on special clinically relevant information with regards to the operation of the myocyte under various conditions, where we will again focus on Cori cycle, and you will be introduced to the alanine cycle as well as how a muscle functions under different working conditions. So let us start by first defining gluconeogenesis. As the term implies, gluconeogenesis is the process of synthesizing glucose or glycogen from non-carbohydrate precursors. That means it has to be a precursor molecule that is non-carbohydrate. So do not get confused if somebody asks you if the process of synthesis of glycogen from glucose, process of synthesis of glucose from glycogen is gluconeogenesis. The answer is no, because the process of synthesis of glucose from glycogen is known as glycogenolysis, and moreover, glycogen is not a non-carbohydrate precursor. So you have to have a non-carbohydrate precursor in order to initiate the process of gluconeogenesis. So let me reiterate the definition again. Gluconeogenesis is the process of synthesizing glucose or glycogen from non-carbohydrate precursors. And the major substrates for gluconeogenesis are glucogenic amino acids, which we will talk in detail when we cover amino acid metabolism, lactate, glycerol, and propionate. Now, lactate, we already know that under anaerobic conditions, we produce lactate from pyruvate. Glycerol, on the other hand, is produced by the metabolism of triacylglyceride or triacylglycerol. And propionate is derived from the metabolism of fatty acids. So the key take-home message here is that gluconeogenesis will use non-carbohydrate precursor molecules for the synthesis of glucose. Now, liver hepatocytes and the kidney are the major gluconeogenic site. But the small intestine may also be a source of glucose in the fasting state. But the major site for the process of gluconeogenesis are the hepatocytes or the liver, and this you should know. So what is the site, or which is the organ that is involved predominantly in the process of gluconeogenesis? The answer is the liver. Now, as I told you, gluconeogenesis, if I want to look at the pathway, is the reverse of glycolysis. In glycolysis, we start the process with glucose, and in the process, glucose is oxidized to pyruvate through 10 successive steps. In gluconeogenesis, we do the reverse, except specific steps in glycolysis have to be reversed, and these steps I am highlighting using the red arrow. So if you can see here, there are 3 steps that require reversal in order for gluconeogenesis to happen. One is the conversion of pyruvate to phosphoenolpyruvate. Second is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. And third one is the conversion of glucose 6-phosphate to glucose. The other steps are in general reversible. Therefore, in gluconeogenesis, you do not require any kind of special mechanism in order to reverse the other 7 steps of glycolysis. So gluconeogenesis is the reverse of glycolysis, except special mechanisms are involved in the reversal of 3 steps. The first one is the conversion of pyruvate to phosphoenolpyruvate. The second one is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. And the third one is the conversion of glucose 6-phosphate to glucose. So these are the steps which involve specific or special mechanisms for reversal. And our job in this particular lecture will be to look at these 3 steps. I will not be going through the other steps, which are simple reverse steps of glycolysis. And the enzymes involved are the same. So for example, the conversion of 2-phosphoglycerate to 3-phosphoglycerate takes place by phosphoglycerate mutase, because this particular reaction is reversible and depends on the availability of the products in either direction. Three glycolytic reactions that I mentioned in the previous slides are essentially irreversible, and the process of gluconeogenesis involves reversing these 3 irreversible reactions. And these are catalyzed by hexokinase, and by now you should be familiar with glucokinase, which is present mainly in the liver and as well as the pancreatic cells. The second one is catalyzed by phosphofructokinase, which happens to be the rate limiting step of glycolysis. And the third one is catalyzed by pyruvate kinase, which is the conversion of phosphoenolpyruvate to pyruvate. So these 3 steps involve specific mechanism for reversal in the process of gluconeogenesis, as these 3 steps are irreversible steps of glycolysis. Now, if I want to get a common mechanism by which the reversal process takes place for the 2 steps that are catalyzed by hexokinase and phosphofructokinase in gluconeogenesis, they involve 2 simple hydrolysis reactions. So by simple hydrolysis, you can reverse the step that is catalyzed by hexokinase and phosphofructokinase. And let us first look at these two reversals involving simple hydrolysis reactions. Now, in glycolysis, what we do is we take glucose and we convert glucose to glucose 6-phosphate, and this process is catalyzed by hexokinase in the tissues and glucokinase in the liver, and the phosphorylation involves the participation of one molecule of ATP that is converted to ADP in the process. The product of the reaction that is catalyzed by these enzymes is glucose 6-phosphate. So gluconeogenesis you have to reverse. That means you need to synthesize glucose from glucose 6-phosphate, and the enzyme that catalyzes this particular step is glucose 6-phosphatase. As I mentioned previously, it's a simple hydrolysis reaction. In the process, we use water and inorganic phosphate is released from glucose 6-phosphate, which is then converted to glucose. So glucose 6-phosphatase reverses the step that is catalyzed by hexokinase or glucokinase. So if I want to write them down, hexokinase or glucokinase during glycolysis convert glucose in the presence of ATP to glucose 6-phosphate, and one ADP is produced. Glucose 6-phosphatase, using a water molecule, carries out a hydrolytic reaction that converts glucose 6-phosphate to glucose, and one inorganic phosphate molecule is released. So we have been able to reverse the step of conversion of glucose 6-phosphate to glucose using the enzyme glucose 6-phosphatase. Now, very briefly, we have to look at how the enzyme glucose 6-phosphatase is regulated. Now, I told you in the class, anything that is increasing the levels of blood glucose will be favored by glucagon, which is a hormone that exhibits properties opposite to that of the hormone insulin. So glucose 6-phosphatase is transcriptionally regulated by factors that call for increased production of glucose. So the first factor that will stimulate the production of glucose 6-phosphatase is low blood glucose. And the second factor that will stimulate the production of glucose 6-phosphatase is glucagon. I'm not going to cover how glucagon mediates its effect because you have hormonal control of metabolism that will be separately taught by Professor Bayumi, and in that particular session he will cover this specific aspect. So reiterating, glucose to glucose 6-phosphate is catalyzed by glucokinase or hexokinase, whereas in gluconeogenesis, the conversion of glucose 6-phosphate to glucose involves a simple hydrolysis reaction that is catalyzed by the enzyme glucose 6-phosphatase, which is under tight regulation by glucagon as well as by the levels of glucose in the blood. We come to the second reaction that requires reversal during gluconeogenesis. In order to do so, let us first look at the normal reaction during glycolysis. In glycolysis, fructose 6-phosphate is catalyzed to fructose 1,6-bisphosphate by the enzyme phosphofructokinase. And this particular enzyme catalyzes the rate limiting step of glycolysis because this specific conversion is the slowest step in the process of glycolysis. Phosphofructokinase, in order to convert fructose 6-phosphate to fructose 1,6-bisphosphate, requires one ATP, which is converted to ADP in the process. Now, in gluconeogenesis, our idea is to convert fructose 1,6-bisphosphate to fructose 6-phosphate. As I told you previously, it's a simple hydrolysis reaction which is catalyzed by fructose 1,6-bisphosphatase, which in the presence of water removes one phosphate from fructose 1,6-bisphosphate, leading to the production of fructose 6-phosphate, and inorganic phosphate is released in the process. So a simple hydrolysis reaction mediated by the enzyme fructose 1,6-bisphosphatase. So let us look at it again. In glycolysis, phosphofructokinase uses fructose 6-phosphate as the substrate. In the presence of ATP, converts fructose 6-phosphate to the product fructose 1,6-bisphosphate. ATP is converted to ADP during the reaction. When reversing this step in gluconeogenesis, fructose 1,6-bisphosphate by the catalytic activity of fructose 1,6-bisphosphatase is converted to fructose 6-phosphate and an inorganic phosphate is released. The process involves hydrolysis, and that's the reason you require one molecule of water. Simple hydrolysis converts fructose 1,6-bisphosphate to fructose 6-phosphate. How is this particular enzyme fructose 1,6-bisphosphatase regulated? Now the take-home message here is fructose 1,6-bisphosphatase is regulated by the levels of adenosine monophosphate. So I mentioned this in the class and we had a note on this. ATP is converted to ADP and ADP is converted to AMP. Now, so adenosine triphosphate to adenosine diphosphate to adenosine monophosphate. So the process of conversion of fructose 1,6-bisphosphate to fructose 6-phosphate is strongly inhibited by high levels of adenosine monophosphate. And the question is, why? I have written the answer here. The enzyme that catalyzes this reaction, that is fructose 1,6-bisphosphatase, is allosterically inhibited and strongly inhibited by AMP. And why? Because when the cell's supply of ATP is low, you already know that AMP levels will be high. The ATP requiring synthesis of glucose from non-carbohydrate sources, that is gluconeogenesis, requires to be shut down. You require to produce more ATP, which means glucose has to be broken down into pyruvate, and pyruvate is then converted to acetyl coenzyme A, which is converted to carbon dioxide and water, and this involves the glycolytic reaction. the TCA cycle, and the electron transport chain, which lead to the production of ATP. So when ATP levels are low or AMP levels are very high, the process of synthesis of glucose from non-carbohydrate precursors will slow down, because such a process, that is gluconeogenesis, requires a lot of ATP, and we shall see that in this lecture, towards the end of this lecture to be specific. So high levels of AMP allosterically inhibit fructose 1,6-bisphosphatase, which is responsible for the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate, and the reaction is a simple hydrolysis reaction requiring the breakage of 1-phosphate from fructose 1,6-bisphosphate. So you have a basic idea about the regulation of fructose 1,6-bisphosphatase. Of course, there are factors such as levels of various gluconeogenic hormones which control this enzyme. And again, I will reiterate, this is not the scope of this lecture. This will be separately covered by Professor Bayumi when he talks about hormonal control of glucose metabolism. So we have bypassed 3 steps which involved simple hydrolysis reaction. So we come to the 3rd step. And the 3rd step requires some elaborate mechanism in order to be bypassed. Let us look at the glycolytic reaction. In glycolysis, we convert phosphoenolpyruvate to pyruvate, and this particular process is catalyzed by pyruvate kinase. We generate 1 molecule of ATP at this state. Now, pyruvate under anaerobic conditions will be converted to lactate. However, under aerobic conditions, it will diffuse from the cytosol into the mitochondria, where pyruvate will be converted to acetyl coenzyme A, and this acetyl coenzyme A then goes into the TCA cycle. Pyruvate is also transported by symport inside the mitochondria by the catalytic activity of the enzyme pyruvate translocase, and once pyruvate comes in, the fate of pyruvate is to be converted to acetyl coenzyme A by the modular enzyme complex known as pyruvate dehydrogenase. So you are well versed with these reactions which are occurring. Now the thing is, I require to synthesize glucose from non-carbohydrate precursors, and the process we now need to follow is gluconeogenesis. So what will happen is we have to reverse this particular step, that is the conversion of pyruvate to phosphoenolpyruvate. Unfortunately, we cannot reverse this step by a simple hydrolysis reaction, unlike the other 2 steps that we looked at. So the major reason is pyruvate, once it is formed under aerobic conditions, is immediately transported inside the mitochondria, except in specific cells where mitochondria is absent, for example, the erythrocytes. But under normal conditions, in normal cells or in a typical cell, pyruvate will be transported to the mitochondria. So what will happen to this pyruvate and how it will be converted to phosphoenolpyruvate? So the first reaction is the conversion of pyruvate to oxaloacetate, and you're already aware what oxaloacetate is, and it's important with regards to the metabolism of fats. So pyruvate is converted to oxaloacetate, and this particular process is catalyzed by an enzyme known as Pyruvatecarboxylase. And this particular conversion from pyruvate to oxaloacetate requires the investment of one ATP molecule, which is converted to ADP. So the first step, pyruvate carboxylase converts pyruvate in the presence of ATP to oxaloacetate. Now, once this oxaloacetate is formed, it is converted to Mallate. And you remember the last step of citric acid cycle, which is catalyzed by Malate dehydrogenase, and it's a reversible reaction. So Malate, so I will write this down here, so bear with me please. So Malate is converted to oxaloacetate in the last step of TCA cycle by the catalytic activity of the enzyme Malate dehydrogenase. I'm just writing DH. When you take down this note, please write the full enzyme. So this is the last step of TCA cycle. And the enzyme that catalyzes the conversion of malate to oxaloacetate is malate dehydrogenase. This is a reversible reaction. So the oxaloacetate that is formed from pyruvate is converted to malate by the enzyme malate dehydrogenase. This malate that is formed inside the mitochondria comes into the cytosol by a transporter, and once malate is in the cytosol, malate is converted back to oxaloacetate by cytosolic malate dehydrogenase. So malate dehydrogenase has two forms, a cytosolic form and a mitochondrial form. Remember that what is inside the mitochondria is compartmentalized inside the mitochondria and it cannot come out. So once malate is transported in the cytosol, it is converted to oxaloacetate again by malate dehydrogenase, but this is cytosolic form of malate dehydrogenase. Once oxaloacetate is formed, it is converted to phosphoenolpyruvate by the enzyme phosphoenolpyruvate and this particular process requires 1 GTP, which is converted to GDP in the process of this conversion of oxaloacetate to phosphoenolpyruvate. Once phosphoenolpyruvate is formed, it goes into gluconeogenesis and goes reverse of glycolysis. That means phosphoenolpyruvate will now move up to form glucose, except at the steps of fructose 1,6 bisphosphate and glucose 6-phosphate, which will be reversed by simple hydrolysis and phosphatases that we have already seen. So let us look at this particular pathway again. Pyruvate is transported inside the mitochondria where it is converted to oxaloacetate by the enzyme Pyruvatecarboxylase. And this is also very important. We talked about this enzyme in the class when we were talking about the specific statement, fats burn in the flame of carbohydrates. So this is a general reaction that converts pyruvate to oxaloacetate. Once oxaloacetate is formed, it will be converted by malate by mitochondrial malate dehydrogenase. Once malate is formed inside the mitochondria, it is transported out from the mitochondrial matrix inside the cytosol. In the cytosol, malate is converted to oxaloacetate by cytosolic malate dehydrogenase, and oxaloacetate is then converted to phosphoenolpyruvate in the presence of GTP by phosphoenolpyruvate carboxylase. Once phosphoenolpyruvate is formed, it can now move into gluconeogenesis. So, as I told you, the reversal of the step of pyruvate to phosphoenolpyruvate requires an elaborate mechanism. This particular aspect that we talked about, that is oxaloacetate getting converted to malate, and then malate coming out or shuttling out from the mitochondrial matrix inside the cytosol, is called the malate oxaloacetate shuttle. In summary, I can say that the reversal of pyruvate to phosphoenolpyruvate requires or involves the malate oxaloacetate shuttle. Very important for exams. Please look at the reactions very carefully, and if you have queries or questions, do not hesitate to write to me. Some facts that you need to know. Pyruvatecarboxylase is an enzyme that uses biotin as a prosthetic group. You already know what prosthetic groups are, so I'm not going to talk about prosthetic group, but this particular enzyme requires biotin as a prosthetic group. And if you look at the process which we looked at it in the previous slide, pyruvate is converted by the enzyme Pyruvatecarboxylase to oxaloacetate, and in the process you require biotin as well as a ATP molecule. So dietary deficiency of biotin leads to hypoglycemic conditions. The other aspect that we have is avidin, which is a protein that is found in eggs, binds to biotin with high affinity. So this is avidin, a protein that is found in eggs, and if you see here, avidin binds to biotin, and the affinity between biotin and avidin is very high. So one particular avidin molecule is able to quench 4 biotin molecules, if you can see here in this figure. So eating or excess consumption of raw eggs leads to biotin deficiency, which may lead to hypoglycemia. So eating of raw eggs is always unadvisable, and the reason is avidin in eggs will quench biotin, and since biotin is one of the key prosthetic groups for pyruvate carboxylase, it will interfere with the process of gluconeogenesis. As a result, the person will have low levels of blood glucose or will have hypoglycemia. Now, a little bit on the regulation of pyruvate carboxylase. Pyruvate carboxylase is an allosteric enzyme that is regulated by acetyl coenzyme A. So why it is regulated by acetyl coenzyme A? Now, when gluconeogenesis is active in the liver, that is, we have increased oxaloacetate directed into gluconeogenesis for the production of glucose, this acetyl coenzyme A should not take up all that oxaloacetate to move it into the citric acid cycle. Recall that the first step of the citric acid cycle or the TCA cycle is oxaloacetate plus acetyl coenzyme A, which leads to the formation of citrate by the catalytic activity of the enzyme citrate synthase. So this oxaloacetate is available, and this buildup of acetyl coenzyme A helps in the use of this oxaloacetate for the formation of phosphoenolpyruvate, and the way that it helps in the formation of phosphoenolpyruvate is by the activation of Pyruvatecarboxylase. There is a detailed regulation of this particular this particular enzyme by hormones, especially by insulin, glucagon, and thyroxine, that you need to look at. And this will be dealt in the lecture by Professor Bayumi. Simply, we do not have the time, neither this lecture has the scope for dealing with these particular regulations. Why these regulations are important? Because under certain diseased conditions, and mind it, and I will mention this here, disease conditions doesn't only mean diabetes, but also situations like hyper or hypothyroidism. You have problems in the regulation of these enzymes, which lead to either decrease or increase in the level of blood glucose. So please ask Professor Bayumi to talk on this, for talk about the hormonal regulation of glucose metabolism. Very important for your exam, and very important not only for your exam at MBRU but also elsewhere. Now let us look at the energetics of glycolysis and gluconeogenesis. Of course, the energetics of glycolysis you already know very well. We will look at the energetics of gluconeogenesis, and then we will try to see if both glycolysis and gluconeogenesis are running together, if we gain or we have a deficit of ATP. In glycolysis, the idea is to convert 6-carbon glucose to 2,3-carbon pyruvate. The process involves 2 NAD plus, and you have a net gain of 2 ATP from the process of glycolysis. I mention here again, we have a net gain of 2 ATP, although the total production of ATP is 4, but we use 2 ATP at 2 steps for the phosphorylation of the substrates. In gluconeogenesis, we have 2 pyruvate molecules that we start with. We end up producing glucose, and in the process we require 4 ATP molecules and 2 GTP molecules. So 4 plus 2, 6 high energy phosphate bonds. So 6 high energy phosphate bonds are broken, which can be written as 6 ATP molecules. So I'm just, for the purpose of calculation, let us say I'm converting everything to one specific unit of ATP. So in the process of glycolysis, we yield 2 ATP molecules, or we have a net gain of 2 ATP molecules. In the process of gluconeogenesis, we spend 6 high energy phosphate bonds, so 6 ATP molecules. So if both glycolysis and gluconeogenesis are running side by side, we call that a futile cycle. A futile meaning we will not gain any ATP. On the other hand, we will have a deficit of 6 minus 2, 4 ATP molecules if these two pathways are occurring side by side. So that's a futile cycle. Futile meaning I will not be generating any high energy phosphate bonds. On the other hand, I will be using quite a bit of energy. So this is a key aspect when both gluconeogenesis and glycolysis run side by side. But the question is, do they occur side by side? And the answer is yes. And we will see certain conditions or certain organ metabolism where these two pathways are occurring side by side. And you are going to answer is, what is the biochemical importance of a futile cycle for me? So I'm not going to mention why they are required to run side by side, but after going through the lecture, you should answer for me the question, why is operation of glycolysis and gluconeogenesis parallelly required in the process of metabolism? In order to explain to you the idea of the futile cycle, I have a new section which is called the section of the muscle story. Now, this is related to organ metabolism. Unfortunately, last year, for some reason, Professor Bayumi didn't cover this part, so I would like to cover it here because you have the knowledge now to look at the muscle story. So, muscle story talks about the operation of the muscle under at different conditions such as exercise, as well as during rest. So this is a plot which talks about exercise time and how the muscle utilizes energy during exercise. Now this plot is not drawn to scale, so this is something that I prepared just to explain to you. So when the muscle starts and workout, let us say somebody starts running, in the first 10 to 15 seconds, the person will use up the ATP stores in the muscle, present inside the muscle cells. So hardly by 40 seconds at most, all the ATP that is present in the muscle will be gone when the muscle goes for exercise. Now, at the same time, the muscle will kick into anaerobic state, and there will be anaerobic glycolysis, and this will be able to sustain the muscle for approximately 5 minutes. Of course, the peak of anaerobic glycolysis stays there for approximately 2 minutes. Now, you have to understand one thing, that when a person is running short distances with high intensity speed, it will depend solely on anaerobic glycolysis and to some extent on the ATP stores present inside the muscle. By proper conditioning training, you can increase both the ATP stores as well as the ability of the muscle to rely on anaerobic glycolysis. And this kind of conditioning you see especially in athletes who are sprinters, so who can run short distances in a very short duration of time. So they have this kind of conditioning of using they have high levels of ATP and are able to sustain anaerobic glycolysis more than even 5 to 7 minutes. But generally, by 2 minutes, you have reached the peak of anaerobic glycolysis. Lactic acid is building up inside the muscle, and at the same time, the other organ systems are catching up with this sudden need for energy. So the heart is pumping huge amounts of blood to the muscle. So as the other organ systems catch up with this need for ATP, we see a gradual increase in aerobic energy system. Of course, the aerobic energy system can also be conditioned, and this is mostly functioning in athletes who are long distance runners. For example, marathon or triathlon runners, they will have a very well developed aerobic energy system to sustain muscle exercise. So that gives you a difference between sprinters and marathon runners. And if you look at sprinters, they are pretty muscular, so their muscles are well developed, they have a large store of ATP, they can sustain anaerobic glycolysis for a longer duration of time, consider in comparison to a normal healthy individual. But on the other hand, people who run long distances like marathon or triathlon runner, they are generally lean, they do not have a highly muscular body, but they have a very well-oiled aerobic energy system which they can use for sustaining muscle activity for prolonged periods of time. So both this anaerobic glycolysis, the aerobic energy system, can be developed by conditioning. So that gives you a fair idea how the muscle will sustain its energy requirements over a period of 5 minutes, where initially for 40 seconds it can sustain it from its normal ATP stores. For the next 2 minutes or so, the anaerobic glycolysis will be able to help it to sustain its activity. And for longer durations, of course, the muscle will always depend on the aerobic energy system. Now let's take a muscle which is resting. So I gave you the example of a person who goes out for a run, comes back home and had a standard American breakfast, a breakfast that contains balanced amounts of carbohydrates, proteins, and fat. And then he is resting. So resting muscle fibers typically take up fatty acids from the bloodstream. Inside the muscle fiber, the fatty acids are oxidized, and the process of oxidation of fatty acids, we will talk about this. Do not worry. So we have something called the beta oxidation of fatty acids. So the fatty acids are oxidized in the mitochondria to produce acetyl coenzyme A. And you are already familiar with what acetyl coenzyme A is. And several molecules of NADH and FADH2. Of course, acetyl coenzyme A immediately enters the Krebs cycle, that is the aerobic energy system, leads to the production of carbon dioxide, ATP, NADH, FADH2, and oxaloacetate. NADH and FADH2 will enter the electron transport chain in the inner mitochondrial membrane, and they will lead to the production of ATP. And you know the balance sheet that you have already done for aerobic metabolism, so you can picturize that when you go through this particular material, either on the video or on the LMS. Now let us move from resting to working muscle. As one begins to exercise, she or he almost immediately uses the ATP stores, that is the ATP that is present inside the muscle, and there is a creatine phosphate system that functions in the muscle. But just to give you a very basic idea, there are basal stores inside the muscle, but by 40 seconds these stores are completely exhausted, and the functioning of these stores needs or involves a particular molecule called creatine phosphate. That's the reason when I wrote it here, I wrote ATPCP. So ATP creatine phosphate system that sustains the muscle for approximately 30 or 40 seconds. As I mentioned there, I wrote this particular message for you. This system dominates in events such as the 100 meters dash or lifting weights. So 100 meters dash, you require a short but high spurt of ATP. And lifting weights, there is a particular event in the Olympics, you may have watched them. It's called the snatch. For snatch, you have to snatch the weight from the ground and pick it up over your shoulders, and some people will be lifting over 200 to 300 kilograms of weight with ease. Such short bursts of energy requirement are sustained by the basal levels of ATP present inside the muscle cells. And by conditioning, of course, you can develop these stores of ATP inside the muscle. Now, after the ATP-CP system is exhausted or depleted, and this particular system is called the phosphagen system, the muscle must find another source of ATP. And the process of anaerobic metabolism can maintain ATP for about 45 to 60 seconds. Conditioning can increase it to further to 2 or 3 minutes, but in general it can sustain ATP requirement for 45 to 16 seconds. What happens is muscle glycogen is converted to glucose by the process of glycogenolysis. We have not looked at this process, but we will talk about it next week. So glycogen is converted to glucose, glucose to pyruvic acid. Pyruvic acid is then converted to lactic acid, and you already know that this process is called anaerobic glycolysis. Now, what is the fate of lactic acid? Many of you asked, and I told at that time that we will talk about this separately, because lactic acid then diffuses out of the muscles into the blood. In the blood transports it to the hepatocyte. The hepatocytes convert lactate back to glucose by the process of gluconeogenesis, which you now know. The produced glucose comes back to the blood and is taken by the muscle again. So as I told you there, I have written a comment here. It usually takes time for the respiratory and cardiovascular systems to catch up with the muscles and supply oxygen for aerobic metabolism. So that's the reason for 45 to 60 seconds, the anaerobic system helps the muscle to sustain its energy requirements. So lactic acid is produced in the muscle, it comes in the blood, from the blood to the hepatocytes. Lactate is then converted to pyruvate, pyruvate to glucose by gluconeogenesis. Glucose comes out of the hepatocyte, back into the blood, carried to the muscle, and the muscle then uses it for production of ATP and lactic acid, and this lactic acid again goes back into the blood, and the process continues. Let us look at this process in a bit more pictorial fashion. So we have glucose. This glucose can be derived from glycogen, or this glucose can be glucose from the blood. So the source of glucose may be glycogen, which by glycogenolysis produces glucose. So I will just add this in this particular figure, which is missing. So glycogen by glycogenolysis gives rise to glucose. Glucose then leads to the formation of pyruvate by the process of glycolysis, because the respiratory system and the cardiovascular system haven't caught up to the rate at the which muscle requires energy. Therefore, the muscle will sustain its energy requirement anaerobically. So pyruvate is then converted to lactate. Lactate comes inside the blood, taken up by the hepatocytes, where lactate is converted to pyruvate. Remember the enzyme lactate dehydrogenase. Pyruvate is then converted back to glucose. In the process, it requires 6 ATP molecules. Glucose then comes into the blood, goes back inside the muscle, and the process of glycolysis continues. This particular cycle is popularly known as the Cori cycle. So don't get confused if somebody asks you what is the Cori cycle. Cori cycle is exactly what I have drawn here or what I just described here. So thus lactate formed by glycolysis in skeletal muscle and erythrocytes is transported to the liver and kidney where it reforms glucose, which again becomes available via the circulation for oxidation in the tissues. The process is known as Cori cycle. So that's the definition and a short description of Cori cycle. Look at it here. You produce 2 ATP, but you use 6 ATP. So it's a futile cycle. But why do you think the body is involved in operating a futile cycle in the Cori cycle? So you need to answer this question for me. So we have talked about a muscle which was at rest. Now it is exercising a lot. So the other cycle that last year was covered in the later organ metabolism, and that's the reason I thought this will be a good place to refer to, that was the glucosealanine cycle. And this is also very, very important for your exam, not only at MBRU but elsewhere. So in general, muscles produce pyruvate from glycolysis during exercise, and this particular amine is produced from normal protein due to degradation of alanine. So when there is degradation of protein, you produce amine groups or amine. But this we will look at when we talk about amino acid metabolism. For the time being, please try to grasp that proteins, when they are metabolized, will lead to the production of NH2 because proteins are nitrogen containing compounds. Now this amine that comes out in the presence of pyruvate is converted to alanine. Okay, so pyruvate, when in the presence of amine, is converted to alanine. You know what alanine is? It's an amino acid, and there is an enzyme involved in the process. We will talk about this enzyme when we come to amino acid metabolism. At this stage, just try to grasp that pyruvate and amine lead to the formation of alanine. Now this alanine comes out of the muscle and through blood is taken to the liver. Now liver converts alanine back to pyruvate and pyruvate back to glucose. You already know that liver is the site of gluconeogenesis. So once this alanine reaches the liver will use alanine as a substrate for producing glucose. Non-carbohydrate, so it's gluconeogenesis. In the process, this amine group is released out of alanine. Pyruvate is formed. So here we combine pyruvate with alanine. Here we take away the amino group from alanine and we get back pyruvate. This pyruvate enters gluconeogenetic process and leads to the formation of glucose. And the newly formed glucose is transported to muscle to be used for energy again. Just like the Cori cycle, where we were using lactic acid, here we are using alanine. Let us look at this process again. So breakdown of proteins lead to the formation of amine. Amine combines with pyruvate, leads to the formation of alanine. Alanine comes inside the blood, is carried by the blood to the liver. The amine group is taken off. Alanine gets converted to pyruvate. Pyruvate then undergoes gluconeogenesis. Once it undergoes gluconeogenesis, it leads to the production of glucose. Glucose comes inside the blood and comes back inside the muscle where it is used for energy production by the process of glycolysis. So this is a simple cycle like the lactic acid cycle, but instead of lactic acid here we are using the amino acid alanine. Now the question that is often asked is what happens with this amine group? This amine group is then used for the production of urea by the process of urea cycle, which we have to talk when we talk about amino acid metabolism. So this particular amine group is converted to urea. Urea is then excreted by the liver and excreted by the urea is just transported from the hepatocytes from the liver to the kidneys where it is excreted out. So that's the fate of the amine group that was combined with pyruvate to form alanine. Now you can see here this is also a way the muscles can get rid of amines because it helps them to excrete out, and in the process they are using it to generate glucose, which is then used by the muscle for production of energy. This particular cycle is popularly known as the GlucoseAlanine cycle. So the Cori cycle is glucose lactic acid cycle, and the GlucoseAlanine cycle is with amino acid alanine. So also you saw how the muscle functions under different working states. Very, very important, guys. If you haven't understood any part of that, the story, muscle story, please come back to me. That's actually one way to integrate the different metabolic pathways. So you are now looking at glycolysis, TCA cycle, electron transport chain, gluconeogenesis, the glucosealanine cycle, the glucose lactic acid cycle, all working together under different working states of the muscle. And this is very, very, very important for you to understand, because all these metabolic cycles function in a synchronous way such that homeostasis is maintained. Before we finish, I wanted to talk about something called the Warburg effect. What is the Warburg effect? It's the Cori cycle which is operating in the cancer cell. Now, in order for a tumor to develop, the cells are, especially in a malignant tumor, the cells are dividing at a very high pace. And sometimes the tumor will not have proper angiogenesis. That means the development of blood vessels to carry oxygen and nutrients to the cells that are dividing so rapidly at a particular locale. So what will happen at this state is that these cells, they want to get energy, but since there is no oxygen coming, or there is a deficiency of oxygen, you can see here, this is the normal cell, this is the cancer cell. The transport of oxygen in the cancer cell is feeble because blood vessels have not grown properly. So what is going to happen here is, in the absence of oxygen, you cannot have aerobic metabolism. The cancer cell then will sustain itself by anaerobic metabolism. So glucose will be converted to pyruvate, pyruvate to lactate. In the process, there is a net gain of 2 ATP molecules, but the lactate is building up in the cell. It is then taken from the cell to the liver, where it is converted back to glucose, and then glucose comes back to the cancer cell and it sustains the growth of the cancer cell, or the rapid division of the cancer cell. This particular effect that is observed in the cancer cell is called the Warburg effect. And if you look at this process, because you are running a futile cycle so many times over and over again, in the muscle, of course, we run it for some time till we switch to aerobic metabolism. But here it will be running indefinitely, because as the cells keep on dividing, more and more lactic acid will be produced, and more lactic acid has to be converted to glucose by gluconeogenesis. Since we are producing only 2 ATP, but in order to produce that 2 ATP, we are using up 6 ATP. The cancer patient starts losing weight and becomes tired. So that's one of the reasons you will see that most of the people who are in their late stages of cancer, they are often asked to rest and have a good meal. Although sometimes with the drugs that they are on, it's not possible to do so, but this is the reason they are mostly weak and they start losing weight. So that's what is called the Warburg effect. Remember, if you do not remember the process or somebody asks you what the Warburg effect is, you can just say Cori cycle in the cancer cell. So we finished gluconeogenesis, pretty simple stuff, but I wanted to focus more on muscle metabolism that is mostly related to gluconeogenesis, and last year there was a delay in covering that specific topic. Next we will look at pentose phosphate pathway, but as I mentioned, do not forget to look at the effect of hormones on the different pathways, especially in relation to how these hormones signal the different metabolic pathways. They are required in detail because nowadays drugs affect these signaling pathways, so questions are asked not only at MBRU but elsewhere as well. With that, I finish this particular session on gluconeogenesis. Have a nice day and thank you for listening.